Today, we're going to be looking at human genetic variation by investigating Alu Insertion Polymorphism in the PV92 region of chromosome 16.
Alu elements are approximately 300 base pair repetitive sequences that have propagated throughout the human genome over evolutionary time. Presence or absence of this element at a specific locus represents heritable polymorphism that contributes to genetic diversity among individuals.
To obtain genomic DNA, we perform a silene malference to collect buccal epithelial cells. These cells are suspended in solution and are used as our DNA source. We then use a centrifuge to pellet the cells, concentrating them from DNA preparation. After centrifugation, the denser buccal epithelial cells form a pellet at the bottom of the tube, while the liquid above, called the supernatant, contains saline and cellular debris and is carefully removed.
We prepare the DNA sample using histogene matrix, which binds divalent cations that could otherwise degrade the sample during heating. We then incubate the sample in a dry bath to lyse the cells and prepare the DNA for amplification.
We amplify the PV92 locus using polymerization reaction or PCR in a thermocycler, which precisely controls when temperature changes. The reaction includes nucleotide, buffer, primers, PEG polymerase combined in a master mix.
During the denaturation at around 95 Celsius, the hydrogen bonds between the DNA strand break, which means that the single-stranded templates are being produced. During the annealing, the temperature decreases, which allows the primers to bind to the complementary sequences, flanking the PV92 region. This step determines the specificity of amplification.
In the extension phase, at around 72 degrees Celsius, Taq polymerase will synthesize new DNA strands by adding nucleotides to the 3' end of each primer. Each cycle doubles the DNA, resulting in exponential amplification.
If the Alu insertion is present, the amplified DNA is longer. If absent, it's shorter. Heterozygous individuals will produce both fragment sizes.
We separate the amplified DNA fragments by using electrophoresis on an agarose gel. An electric current is then applied, and since DNA is negatively charged, it will go to the positive electrode. Smaller fragments travel farther than larger fragments, allowing for separation of size.
To make the DNA visible, we stain the gel with a DNA binding dye, which allows the fragments to be seen under UV light. Each visible band represents DNA fragments of a specific size. One band might indicate that the DNA is homozygous, while two bands may indicate heterozygosity. These patterns allow us to determine potential genetic variations between the PV92 locus.
This investigation demonstrates how molecular techniques and lab equipment are used to analyze genetic variation, with applications in forensics, ancient studies, and evolutionary biology. It also allows us to calculate allele frequency and genotype frequency and apply that to the Hardy-Weinberg principle. Thank you for learning with us.